My name is Gauri Gajeshwar, and I am a senior in the Duclos Lab at Brandeis University. As a part of the Brandeis MRSEC IRG2, we aim to control far from equilibrium active matter systems such that they mimic biological organisms. What is active matter and why is it important? In active matter systems, individual entities use energy to move and create collective motion. Active matter exists at all line scales and is involved in important biological processes like cell division and morphogenesis. Studying these systems helps us better understand and control our biology. How can we control active matter at a microscopic level? We draw inspiration from the collective movement of this herd of sheep. Sheep from the herd have aligned unidirectional motion to create a collective movement, and their path of movement is directed by obstacles or trees in the landscape. Our goal is to mimic the biological behavior of a herd of sheep on the molecular level. To do so, we will use the microtubule kinesin system as our molecular sheep. Here, rod-shaped microtubules glide on top of a layer of surface-attached kinesin motors. We can direct the motion of these molecular sheep using obstacles. We use MinDE proteins for our molecular obstacles. MinDE proteins interact with each other to make patterns on a lipid membrane. The MinDE protein spots you see here kind of resemble trees in a field. We can combine the pattern-forming MinDE and activity-generating microtubule systems because of a protein called streptavidin. MinDE proteins push streptavidin into complementary patterns. Streptavidin sticks to the motor protein kinesin, and kinesin helps our molecular sheep or microtubules move. Thus, individual microtubule motion can be directed using MinDE protein patterns. At high concentrations, microtubules will form bundles, creating collective motions similar to a herd of sheep. Our current research asks the question, can dense microtubule bundles also be directed using MinDE protein patterns?